Our charge here at the lab is to find ways of maximizing plant productivity. The approach we use to do that is genetic engineering, which is the quickest way you can find chunks of DNA that modify the behavior of a plant and make it more adaptable to drought, more adaptable to a disease, and just produce more biomass per unit area. We have since found out that the majority of plant species do not accept foreign DNA as readily as we would like them to. They are recalcitrant. Our charge is to find ways of overcoming those difficulties. How do we introduce those chunks of DNA into plants of interest? And how do you then regenerate a whole plant that has that chunk of DNA and that then subsequently performs at industrial capacity? The technology that we've come up with here at Oak Ridge National Lab is we've identified 7 genes that regulate cell cycle, cell division, cell multiplication, and those multiple cells then are reprogrammed to form a shoot tissue and a root tissue, to where now you have an entire little plant that you can then take out and then plant actually out in the field, and then you have your whole industry going from there. The first mandate for us, we need to make sure that actually this technology works for plants that are dedicated to biofuels and bioproducts. And we have demonstrated that under natural systems, this works in poplar. And so now we use a model system, which is Arabidopsis, to see can we replicate that behavior, that genetic behavior we saw in poplar in Arabidopsis. We are also working with sunflower to see if the behavior is conserved across multiple plant species. And then from that point, then we know we have found a broadly applicable technology that we can then apply to all these plant systems with no limitation. And so we see this technology as enabling industries in terms of we can now make plants adaptable to their production environments. We can make them more productive by using these genetic engineering techniques.